Hi everyone. Today I would like to go ahead and give you a quick overview of Chapter 11 and 12. Both of these will detail the nervous system. I want to emphasize that this is an overview. It is meant to complement the chapter outlines that have been posted for you on Canvas. Please make sure that you read through those. And of course, after listening to the overview and reading the chapter outlines, if you have any questions, comments, or concerns, please do not hesitate to contact me. So let's go ahead and take a look at Chapter 11. It is entitled Fundamentals of the Nervous System and Nervous Tissue. And it is our first chapter in our text that will start to introduce us to the nervous system. Now, when you think of the nervous system, I think many of us will say, oh, that's like the brain and the spinal cord and the nerves. And you're completely correct in that. But the nervous system as a whole is an excellent way for our body to communicate with internal and external stimuli. And what the nervous system then brings to the table is a way not only to receive that sensory information, but also to figure out the integration, meaning once you receive the message, what is the action that needs to be done, and how do we then send or employ that particular mechanism of action so that we can get a result. So the brain is that perfect in between of receiving a sensory input, integrating or analyzing the information, and then deciding on what the output or the result is going to be. About the nervous system, we can roughly divide it into two principal parts. So we have what we call the central nervous system and we have the peripheral nervous system. Very important to highlight that. The central nervous system basically takes care of the brain and the spinal cord, and it's often abbreviated as CNS. And then anything that is nervous system related that is outside of the brain and the spinal cord, such as when you discuss your cranial nerves, your spinal nerves, or your ganglia, that then falls under the peripheral nervous system or PNS. Now, ganglia are just clusters of cell bodies of the neuron. And when the neurons bundle together and elongate and leave the spinal cord, we then refer to them as nerves. So you can see that you have cranial nerves, which originate from the cranium, and then we have our spinal nerves, which will originate from the spinal cord. In one of our future chapters, we're going to go ahead and name them so that we can explore how each of them contributes to the overall sensory and output system. Now, on our next slide, we're able to see how the nervous system is completely organized. And as you can see, starting on top, you have your central nervous system and your peripheral nervous system. And I want you to think of the central nervous system as the control center. So this is like where the integration, where the sensory system is going to ultimately lead you to. This is where we're going to figure out what is happening and what needs to be done. And it's the control center. So that's the brain and the spinal cord. And the peripheral nervous system, so your cranial and spinal nerves, your ganglia, those are the ones that are going to be able to receive the signals, or I should say, set up the signals to send over to the central nervous system. And once the central nervous system has figured out what to do or respond to, the peripheral nervous system will be the one that will actually do the task of completing the output. So notice how you have blue and red arrows indicating the flow of information. And as you can see, it's a constant communication between the central nervous system and the peripheral nervous system. Now, peripheral nervous system, since it has such a large job, you're going to see that it is subdivided on the sensory and the motor division. And the sensory division, as the name indicates, is going to be all about being able to receive these impulses from receptors and allowing those impulses to be communicated with the central nervous system. Notice how they wrote that it's composed of somatic and visceral sensory nerve fibers. Somatic is going to indicate more of an internal working. Sorry, my apologies. Somatic is going to indicate more of an external working. So we're thinking like our skeletal muscles, whereas the visceral is going to be more of an internal sensory system. So we're thinking as our different organs, our glands, anything that's occurring within the thoracic or abdominal cavities. Now, the motor division will be our efferent division, efferent because it's going to allow for the effects to occur. And the effect can either be a somatic or an autonomic effect. A somatic nervous system effect means that it will go ahead and involve somatic motors. These are voluntary, and that means that it's all about getting the skeletal muscles to respond. So, for instance, if you are walking to class and you see that, or you start to feel that it starts to rain, you'll notice that you can kind of fence yourself to start running. The motion of you engaging your muscles so that you can go from walking to running, that would be an example of the somatic nervous system at work, telling your muscles to contract and relax faster so that you can run and not be in the rain and get soaking wet before your class starts. The autonomic nervous system is going to be all about the visceral motors, and that's basically what's happening on the inside. So, how are we telling the cardiac muscle, the smooth muscle, and the glands to react? And as you can imagine, if we're going to create a visceral reaction, it can be one that can emphasize or it can be one that can calm down. So you notice that under the autonomic nervous system, there is another branching between the systemic division and the parasympathetic division. The sympathetic division is everything that will increase and hype you up. So, for instance, when you're exercising, you feel that your heart rate and your breathing patterns start to increase, and that is because your sympathetic division is being activated. The parasympathetic is the calming down, the restoring back to homeostasis. So, one will increase, that's sympathetic, and the other one will decrease, that is parasympathetic. And they tend to usually work in union together so that we can maintain our homeostatic rate. Now, when it comes to the nervous system, there are two main cells that we have to discuss, and this might remind you of our histology chapter, which we didn't do all that long ago, which discussed that nervous tissue is composed of the neurons and the glial or neuroglia cells. The neurons, of course, are the workhorse. They are the ones that are able to receive signals, send off an effort or an effect. And then the glial cells are going to be the supportive cells. And you may recall that they greatly outnumber the neurons, usually about 1,000 to 1. And the glial cells come in many formations, and each of them have a particular function. So let's kind of talk about the glial cells a little bit. Start off with the astrocytes because they are the most abundant that you will find in the central nervous system. Astrocytes play many different roles, but they're probably best known for the fact that they help regulate the blood-brain barrier within our nervous system. The next one to discuss are the glial cells, microglial cells. Those are an example of defensive cells that we also find within the central nervous system. The epidermal cells are really important because they're going to allow us to produce the cerebral spinal fluid, which we use for the lubrication and the filling of the cavities within the nervous system. And then this one right here, D, this one's called the oligodendrocyte. We're going to talk about that one a little bit more because what an oligodendrocyte does is it does this thing called myelination, which means it forms a myelin sheet, which is mostly made out of lipids. It forms this protective sheet around the axons of the nerves in the central nervous system. And that same process of myelination is also seen in the peripheral nervous system, but it's going to be done by what we call a Schwann cell. So Schwann cells and oligodendrocytes, both of them will do myelination. The main difference is the location of it. The oligodendrocytes will do myelination in the central nervous system, while the Schwann cells will do it in the peripheral nervous system. Helping out the Schwann cells in the peripheral nervous system will also be what we call the satellite cells, which are another example of the glial cells. Here an image of a typical neuron, and what it's trying to highlight is that there's three main areas that you should concentrate on. You have the dendrites, which are the receptive entity, and as you can see, there are multiple dendrites that are all radiating from the central area right here. This is called the cell body or the soma. That is where you're going to be finding your nucleus. And then finally, one of them will have a little bit of a wider extension, and that is because that is the axon hillock, which will give rise to the axon. And the axon is the one that will send off the effect. So as you can see, there's a little arrow right here telling you the impulse direction, and it will end up with its little axon knob. So some of you might be thinking about the neuromuscular junction, and that would be, of course, the tail end of your axon communicating with your skeletal muscle by releasing acetylcholine. So dendrites receive, the cell body integrates and figures out what needs to get done, and then the axon is then the one that sends off the message. And these little white little beads that you see all around the axon, that is the myelination, or as you can see right here, it's the myelin sheath. And the little openings in between are called the nodes of Ranvier. We're going to talk about how the myelination actually helps speed up the electrical signal or the action potential down the axon. And I'll tell you, or I'll show you an illustration of how the myelin sheath can actually increase the speed of that signal versus what we call an unmyelinated axon. Here's an image of a swan cell. And what's happening in here is the little nucleus in purple. As you can see, it's starting to wrap itself around an axon. Now, let me just take a moment and remind you that the swan cell does myelination in the peripheral nervous system. And it will do myelination by starting to wrap itself around the axon until it is loosely wrapped with multiple layers. And that will then create the myelin sheath. And each swan cell will be responsible for its own section of myelination. If you look at the picture on the side, you can see that it creates this nice thick rim of myelin, which mostly is lipid, mostly fat. And this happens during the first few months of after birth. My apologies. So this myelination will start after birth and it will kind of proceed throughout the first few years of our development. And like I said, it's mostly based out of lipids, and its goal is to speed up the chemical and electrical signal of this neurons. The other thing I want to highlight is the fact that the swan cell in the peripheral nervous system has a complementary cell in the central nervous will also do myelination. That is going to be your allegrodendrocyte. So here on the illustration, you can see it. Now, one of the main differences is that the swan cell will have an individual myelination for each of itself, whereas the allegrodendrocyte will have one cell that will branch out and myelinate several sections of axons in the central nervous system. So one cell can myelinate several axons in the central nervous system, and that is the allegrodendrocyte, whereas the swan cell will only myelinate one particular section. So as you look at peripheral nervous system and you see the myelination, you can see that each little bubble, each little nodule is an individual swan cell. Now, the myelination will show up as white matter. So like in a little bit when we talk about cross sections of the brain and the spinal cord, you're going to notice that it's been color coded to indicate what we call gray matter and white matter. White matter are the myelinated sections, and then the gray matter are the cell, like the dendrites, the soma, or the unmyelinated axons. And not everything is myelinated in our nervous system, and the simple reason for it's because of space. We do not have enough space for all of the axons to be myelinated. Table 11.1 is going to go ahead and give a comparison over the different classes of neurons that we encounter. The most popular one is what we call a multipolar neuron, and that basically means that you have multiple dendrites or processes that will extend from the cell body, and you will have one axon that will radiate outwards. Bipolar means that you only have two extensions. So you could see here, you have the cell body towards the middle, and then you have a fusion that represents the dendrites on one side and a fusion that represents the axon on the other side. So bipolar meaning two. And then unipolar, these are actually most common for us during embryonic development. That basically means that you have a cell body that only has one process. You can see only one branching off from the cell body. And as it radiates out, this elongated axon will then have a peripheral process and a central process. Unipolars are sometimes also referred to as pseudo unipolars, and that is because in some cases our body will have a bipolar or a multipolar neuron that will go ahead and alternate its overall shape to become a unipolar. At the bottom section of this particular section of the chart, it shows you the relationship of what they call the receptive region in blue, the conducting region in green, and then the secretory region in red. And it just highlights the fact that you have an area that will receive the stimuli, courtesy of the dendrite, sending it over to the cell body. And then you have your conduction region. This is where your action potential is going to be triggered and falling down or traveling down the axon. And of course, it will get to the end where you then have the secretion of, in this case, the neurotransmitters. So I want us to kind of think about the neuromuscular junction from our previous chapter and realize that as the action potential comes down, it will cause the release of acetylcholine. Acetylcholine, norepinephrine, these are all examples of neurotransmitters, meaning that they have been generated within the nervous system. This particular section, this is a continuation of table 11.1. It now takes a look at the abundance of the particular neurons. And as I mentioned, the multipolar neuron is the most abundant in the body. It is the major neuron found in the central nervous system. Bipolars are more for some special sensory organs, so they are relatively rare. And then the unipolar or pseudo-unipolar, those are found mostly in the peripheral nervous system, within the dorsal root ganglia, the sensory ganglia of the cranial nerves. At the latter part of the table, it talks about the function. So you can see that the multipolar neuron, being the main one within the central nervous system, it will have the role mostly of being what we call an interneuron. And the interneuron basically means that it conducts impulses within the central nervous system. It integrates, it connects the sensory input or the motor output. So the interneuron is your connection between sensory, or my apologies, the interneuron is your connection between the afferent and the efferent, the sensory and the output. Some of the multipolars are also going to be motor neurons. So these means that they are going to allow for the signal to be transmitted along the efferent pathway of the central nervous system overall to what we call the effector, or the part of the body that will respond. So the muscle will contract, the gland will cause a secretion. The bipolar and the unipolar, these are mostly sensory neurons, which kind of makes sense based on their overall location and their action of sending off and receiving different impulses. Now, you can't get away from that action potential. I know some of you wish that you could, but the action potential is also going to be discussed in this particular chapter, because as the nervous system receives sensory input and figures out what needs to get done, the way that the message is going to get sent along the axon is by generating an action potential. So the chapter will really quickly remind you of the main steps, which involves the fact that you would need your sodium and your potassium channels and the movement of these ions in and out of the cell to go ahead and manipulate the membrane voltage that you are exposed to. So in the first illustration, you can see you have a resting state, meaning that both your sodium and your potassium channels are closed. And as you can see, it's at a resting membrane potential. If then we go ahead and proceed to the opening sodium channels and the sodium flowing into the cell, we see that that will cause the membrane voltage to increase. And that, of course, is what we refer to when we say depolarization is occurring. The depolarization will go ahead and cause the peak of the action potential, the peak of the excitement of the cell, at which point what we see happening then is that it's time for the sodium to close off and the potassium to open. Now, potassium ions will flow out of the cell, and as they're flowing out of the cell, they allow for the membrane voltage to decrease, and this, of course, is our repolarization, or as I like to think about it, our reset stage of returning back to the resting membrane potential. Eventually, what we see happening is that there is a slight bit of what we call hyperpolarization. Part of that has to do with the fact that your sodium channels will remain open for a little bit longer, while the potassium channels will remain open for a little bit longer while the sodium starts to set up. Also, that the potassium floods out so much faster that it actually causes the membrane to be slightly more negative than it was for its resting membrane potential. That dip below its RMP is what we call hyperpolarization. So you can see it's a little bit more negative than where it started from. Now, the reason action potential in this chapter, it's not because we're doing muscles. No, we're not. We're taking a look at the axons. As you may recall, some of the things that muscles and neurons have in common is that they're both able to receive stimuli and then react accordingly. And in this case, what we see happening is that within the axon, we can go ahead and once properly stimulated, the action potential can then travel down the axon to indicate the excitement and eventually, or perhaps, cause the release of a neurotransmitter or an electrical signal, allowing for the communication to occur. And what this picture shows you is what we call propagation of an action potential. And propagation basically means that you have a starting point and then you have a road that you follow as the action potential goes from one side to the next. And on the illustration, you could see that they've done different color codes. The yellow is for the resting potential, meaning that it has the potential to be manipulated. So we're talking about sodium channels opening and sodium flooding in to get it nice and excited. In red, you have an actual peak of action potential. So those cells have already made it over to the most excited state that they're going to be. And the sodium channels are just about to close off and allow for the potassium channels to open, which will then cause a sea of gray to occur, and the hyperpolarization is then indicated by that color. Notice how on the membrane that they're highlighting, you have the positive and the negative to indicate the movement in the membrane voltage. And you can see that the action potential in red will go ahead and move from its start to the middle to the end of this particular axon. The movement of the action potential, this is now what we call propagating. And this, of course, is our typical action potential graph. You can see the depolarization, courtesy of sodium entering, and then the repolarization, courtesy of the potassium leaving the cell. Now, this chart also highlights what we call an absolute and a relative refractory period. An absolute refractory period means that as you're looking at the reaction of the cell or the membrane, it is right in the middle of producing an action potential. So no matter how much you increase the stimulus, you will not get that membrane or that cell to produce an action potential on top of the one that is already occurring. So notice in the chart how the absolute refractory period basically entails the height, or I should say the majority of the section of depolarization, action potential, and some of repolarization. And that is because at a certain point of the cell resetting itself, it switches from absolute refractory period to relative refractory period. And relative refractory period indicates that you can excite the cell to produce another action potential, but that would mean that you would need to have a higher or greater stimulus than you would have when the cell is at rest. And part of that has to do with the cell still being in the process of resetting itself. So absolute refractory period, no matter how big you make that stimulus, you will not get a second response. Whereas the relative refractory period says that you can get a secondary response, but you will have to increase the stimulus needed in order to do so. Here is an illustration showing you how the propagation of the action potential can vary in, for instance, dendrites and axons, or more specifically, if you have a non-myelinated and a myelinated axon. Let's start off with just a typical plasma membrane, a bare one, that we would see, for instance, in the dendrites. What we see happening here is that as the stimulus comes in, and it's able to generate a voltage, it's able to generate a response, we see that that voltage will start to decay as it's moving further away from the stimulus. And part of that is because there's no voltage gated channels that can be found. The membrane is what we call bare. So once the stimulus is sent off, what we see is that the response will start to taper off the further away it is from the origin point. Now, in an axon, we see a different scenario because axons have voltage gated channels. So they have access to the sodium and the potassium. So here in this illustration, we once again see that a stimulus is put into play and a response is created. And as the response occurs, we see that it starts to taper off, but it still propagates itself. And part of the reason why the propagation can occur is because you have access to the sodium and the potassium channels in this particular. This is called a non-myelinated axon. Now, why is the conduction so slow and why do you still have a little bit of tapering before it hits the voltage-gated channels? Well, part of that has to do with the fact that it takes time for the ion channels to recognize the voltage and to open up and to allow the movement of the ions through. So as this time is occurring, it slows down the movement of the signal to get from its origin point of the stimulus all the way to the end of the axon. So what can be done to speed up this process? Well, that is where the bottom picture comes into play, and that is what we see when we have a myelinated axon. Now remember, myelination just means that you take a lipid layer and you wrap it around the axon. And it turns out that by doing the myelination, it will allow for the current to keep from its entry point to its exit point of the myelin sheath. So notice how you have your stimulus, you have your large cone of your voltage response, where the voltage is staying relatively consistent until it gets to the gap. And in the gap, we're going to be able to recharge that voltage because look what we have over there. We have our voltage gated channels over there with the sodium and the potassium. So the sodium can go ahead and flood into the cell, allowing for the stimulus, or I should say the size of the voltage, to increase once again. And this only needs to be done in between the myelin. So what we see happening is it almost appears as if the voltage is hopping from one myelin sheath to the next. And this allows it to do a much more rapid speed than if it has to be recharged every single time it comes across a voltage gated channel. So the myelination allows the action potential or the response to literally hop or jump from one section to the next instead of having to be recharged every single channel it encounters. Extremely smart system. Here is a look at a synapse. A synapse is a meeting point between two or more neurons. You can see here that the axon of the presynaptic neuron and the postsynaptic neuron are engaging in their connection. And a fun fact is that a lot of studies have indicated that the level of intelligence is not necessarily related to the amount of neurons that an organism has. It has to do with how many synaptic connections those neurons can make. So just something to think about always engaging your mind to learn as many new things as possible, right? Now, when it comes to that meeting point, you can have several connections. It doesn't always have to be between the axon and the dendrites, although that one is the most common. We call it an axodendritic connection. You also have an axosomatic connection, which is between the axon and the somatic, or the soma, I should say, the cell body of the secondary neuron. You also have cases where you can have axon to axon. We call it axoaxonal, dendrodendritic. Sorry. I'm sorry, I really had to turn my tongue for that one. That is from dendrite to dendrite. And then somatodendritic, that is from soma, cell body, to the dendrite. As these connections are being made, you can either see a chemical release, so we're talking about like a neurotransmitter like acetylcholine, norepinephrine, or you can have an electrical synapse. And an electrical synapse usually means that you have voltage gated movement, or you have like gap junctions that will allow the electrical signal to move from one neuron into the next. Here is a nice little illustration where you can see your axon, and you can see it has different synaptic connection. Here is an axonosomatic, where the axon and the soma are interacting. Here is an exodendritic between the axon and the dendrites. Here are the dendrites. Here's the axons from a secondary neuron. And then here you can see within the axon, you have 1 and 2 neurons interacting. So you have an axonal synapses. On the tail end of chapter 11, it will talk about what we call postsynaptic potentials. And this has to do with the fact that the neurotransmitters, these receptors tend to have a graded potential. A graded potential, unlike an action potential, can vary in the overall strength of its response. An action potential is an all or nothing response. Once you stimulate it properly, you will hit that peak and you will reset. When you do a graded potential, the length, or I should say the amount or the strength of it, will depend on how much neurotransmitter is released, and also the amount of time that it stays within the cleft or the connection point between the neurons. And these graded potentials can have two outcomes. You can have an excitatory response, which we call EPSP, excitatory postsynaptic potential, which means that you get the cell excited, or you can have an inhibitory postsynaptic potential, IPSP, which means that you will calm down the cell or make it less likely for that cell to start depolarizing. Once again, this is only seen in a graded potential. When you do an action potential, your only response is to induce depolarization or excitement within the cell. Here is a look from a graph aspect of an EPSP and an IPSP. So notice that you have the stimulus that comes in, and the stimulus can either cause an increase in membrane voltage or a decrease in membrane voltage. When it causes the increase in membrane voltage, that usually means that we're getting the cell closer to a depolarization state. And part of that is because we're allowing sodium and potassium to pass simultaneously, but they're opening these chemically gated ions. So it's trying to bring the neuron closer to the threshold so it can have a full reaction. In an inhibitory postsynaptic potential, we are trying to inhibit the cell from responding. So we're trying to drive it away from an action potential threshold. We don't want it to react. We want it to calm down. We want it to be inhibited. And this, of course, will not require any sodium, but instead what we see happening is that we're going to rely on potassium or chloride. Either one of these will cause a repolarization step to occur, meaning that it drives it below its resting membrane potential, making harder for an action potential to be induced. I think if we take a look at this table, this is table 11.2, I think we'll get a better understanding of comparing a graded potential and an action potential. So starting off with the graded potential, that one's usually seen within the cell body and the dendrite, whereas your action potential is more about the axon hillock and the axon. Your graded potential, that usually is a short distance, also because of the fact that the further it gets away from the stimulus, the more it slows down and lowers its response. Whereas the action potential, because it's an all or none response, can be utilized for a longer distance. The graded potential, remember, it can vary in its response. It all depends on the stimulus that's being placed upon it, as well as the frequency of the stimulus. It can be a chemical or sensory type of stimulus. And the repolarization is voltage independent. And the reason for that is because it doesn't depolarize and repolarize depending on the sodium and the potassium channels and the action potential. Instead, it basically responds until the stimulus is no longer present, and then it will go ahead and reset itself back in. Action potentials, on the other hand, that is an all or none response. So once you hit that threshold, it will fly up, depolarize, and come on back down, repolarize. It will not decay with distance, and it will, of course, voltage depolarization will be the stimulus to open up your ion channels. And keeping that in mind, that means that depending on the voltage going down, that will then trigger for the repolarization step to occur, meaning that the sodium channels will close off and the potassium channels will open back up. Here's just a look at the fact that you can amplify a graded potential, which you cannot do in an action potential, because the action potential is an all or none phenomenon. You can increase the stimulus in a graded potential by either doing a temporal summation, meaning that you go ahead and you increase the frequency of the stimuli, or you do a spatial summation, which is that you have multiple points of stimuli that are working on the same cell. And keep in mind that with the graded potential, you can have a response that either excites or that inhibits. Okay, whereas action potential, we are only getting excited. Okay, and we are only doing our depolarization, we are only generating our action potential, and we are only constituting that nerve impulse, allowing it to go down the axon. Here's just one last look at the graded potential versus the action potential. This means to highlight the fact that in our action potential, we rely on the opening of the sodium for the depolarization step and the opening of the potassium for the repolarization step. Whereas if we're doing our graded potential, whenever we're doing our excitatory response, we're going to cause the movement of sodium and potassium simultaneously. Whereas if we're doing our inhibitory response, then sodium doesn't come into play at all. Instead, in order to cause a decrease in that membrane voltage, we're going to rely on the potassium and the chloride. All right, so please take a look at table 11.2. I think it does a fantastic job at comparing a graded potential versus our ever so loving action potential. As the chapter, or this is chapter 11, finishes off, it does talk about this thing called a reflex. A reflex is basically an autonomic or a uniform type of response that the body does every time it is stimulated. Most of this is done as a way for us to protect ourselves. For instance, you put your hand on something hot before you can even think about it, you've pulled your hand away. And that's because the minute that stimulus is received, it has the same 5 meeting points that it encounters. So it talks about the fact that the stimulus is detected by the receptor, which is communicating that with the sensory neuron. That will bring it over to the integration center where you have your interneurons, the most common neurons in the body. That will quickly figure out that you might be in harm's way. So it will go ahead and stimulate the motor neuron to cause an effector to be activated, and then you can ultimately get your response. These 5 steps are always followed when we take a look at a simple reflex arch. So the minute the stimulus is detected, it will go into play and you will always get the same uniform response over and over again. Now, the neurons can also form what we call a circuit, which means that they will have more of a collaborative effort as to how they go about sending out their signals. So here on this slide, you have an example of 4 different circuits. Starting with the one labeled A, we have our diverging circuits. This is basically what we'll call an amplifying circuit, where you have one input that is then distributed to many other neurons, allowing for the signal to be amplified and thereby allowing for a greater output to occur than if there was only one neuron creating that. So this is a diverging circuit. The converging circuit is when you have many inputs, but you only have one output. And this really allows for more of a concentrating type of response to occur. Reverberating, that one is like arithmetic activity. It basically means that you have a chain of neurons that are consistently feeding each other, but they're all leading to one output. So if you follow these little arrows, you notice that the neurons are reacting and stimulating with each other, but they're just continuously feeding back to each other and one solo output. This is really like when you take a look at repetitive things like walking, the gait movement of 1 foot forward, 1 foot forward, that really is an example of a reverberating circuit, just in essence creating this chain of neurons that are consistently feeding back on their input and allowing for one output to occur. If you're looking to create kind of like a little burst, then what you need is a parallel after discharge circuit. This is when you have neurons that are arranged in a parallel format that will converge on a single output of a cell. And these impulses will reach the output different times. And that then kind of causes these little bursts of impulses that come into play. So this is just some cool ways that these neurons interact with each other, right? Diverging, converging, reverberating, and parallel after discharge. Now, I know that was a very brief overview of the chapter, but please keep in mind that it's meant to complement the more detailed chapter outline that has been posted on Canvas. Now, let's take a look at chapter 12. And chapter 12 is going to go ahead and focus more on the central nervous system as a whole, taking a look at the brain and how the specific areas will correlate with specific functions for our body. The central nervous system, as a reminder, basically consists of the brain and the spinal cord. Encephalization is when we take a look at the development of the brain and the frontal portion or anterior portion of the body. How do we increase our number of neurons? How do they start interacting and making their synaptic connection? Here we see some of the brain development that starts off with some what they call flexures, which means the folding over that will allow for the anterior, the mid, and the posterior to form. And as you can see, there's quite a bit of development between week 5, week 13, and birth. As you go through chapter 12, you to take your time and I want you to study the different sections of the brain. It is very interesting how they have their brain mappings that will kind of correlate a function of the brain with a specific entity, such as speech, memory, motor function. So you're going to notice that there are lots of illustration in this chapters, just kind of highlighting these particular areas of hearing and reading and talking. This particular slide highlights the distribution of gray versus white matter. Remember, gray matter is what we see with what we correlate with the cell bodies and non-myelinated neurons. The white matter, that is mostly myelinated axons. And remember, since the myelination is mostly lipid, it will go ahead and appear to have that whitish offset. Also, notice how in the spinal cord, the gray matter is in the center while the white matter is on the outer edges. And that pattern is reversed when we take a look at the brain. In the brain, you have the white matter that's in the center, and you have the gray matter that forms the outline of the structure. Speaking of the, when you look at it, you're going to notice that it does have these particular grooves and little channels that are going on. The grooves are what we call gyrus, or gyrus, some people like to say, these little bumps. And then the little fissures are basically the sulcus. Those are like the grooves, these little lines that are generated. So, you can take a look at this picture here at the bottom, where it kind of show you that the gyrus are the bumps, or gyrus, as people like to say, my apologies. And then the sulcus are the infoldings, the little channels that come into play. Part of what the gyrus and sulcus allow is for an increase of surface area within a lobule, so that way we can accommodate more neurons. And remember, it's not necessarily how many neurons you have, synaptic connections between these neurons. All right, let's take a look at this illustration. This is done by completing an MRI. And what we're seeing here is that depending on the activity that the patient is doing, seeing, hearing, speaking, or thinking, it will go ahead and highlight the brain that is most metabolically active by indicating it in a reddish orange tint. So it's very interesting to me how different parts of the brain highlight. So as you can see right here, if we're talking about seeing and visual cues, we're more towards the back of the brain. Hearing will be more associated with the temporal lobe. So most of us are going, that makes sense because that's where my ear is, right? My auditory canal. Speaking is also located more in towards the front. And then thinking is more spatially divided, and part of that has to do with if it's a recall or if it's more like a critical thinking task that occurs. As you're doing this particular section, I want you to take your time because I, for one, am very interested to learn about things like where the particular sensory areas and motor areas are divided within the cortex. So I will kind of talk to you about the differences between a motor cortex, which is more focused on the afferent section of the nervous system, and then, of course, the sensory, which is more afferent and receiving stimuli, and how these two are then able to communicate with each other. We'll also talk to you about body maps, which are just sections of the brain which will correspond with different areas of the body, where you can find within the gyrus, you can find the motor and the sensory system. In fact, if you've ever heard of a homunculus, it is basically a fun activity where you determine the sensitivity of different parts of the body, and then you mathematically try to figure out or you calculate how many neurons are dedicated to that particular section. An area of your body that is extremely sensitive usually means that there are more neurons indicated to that section or dedicated to that section than an area that is less sensitive. Whenever you have some time, go ahead and Google homunculus. I think it is a very interesting read. And in some of the labs, I know in my comparative animal physiology lab, the students will actually go ahead and map out different parts of their body so that they can get a representation of their homunculus. Now, table 12.1, that one is an interesting one. It's going to go ahead and highlight the functions of the major brain regions. And as we start to learn the different sections by name, you're going to utilize the illustration. And you can see it's going to be highlighted. So I won't read verbatim from the table because I know you'll be able to do that. But by all means, take a look at, for instance, the cortical gray matter and how it talks about interpreting sensory inputs and being able to control voluntary skeletal muscle. The thalamus is more of a relay sensory system. For those of us moving on to 2086, we're going to learn a lot about the next section, which is the hypothalamus. The hypothalamus has many functions within the brain. It is our internal thermostat. It helps regulate food and water intake. It is one of the main regions where we go for the endocrine system. It has a boatload to do with our reproductive cycle. So it plays many, many roles within our central nervous system as well as the endocrine system. The lymphic system highlighted here in, looks like an orangey red shade. That one is going to go ahead and play a role in like an emotional response, some memory reprocesses that come into. We have our midbrain, where we have some visual cues that come into play. We also have some central cranial nerves that will radiate down from the pons, which are in the light green section. Those are going to be more of a relay information. And then pons also help with like your respiratory center to help control your breathing rate. The medulla oblongata, yeah, that's the section right here. The medulla oblongata is studied in its sensory pathway impulses to do with the skin and proprioceptors, which are receptors that allow us to um, navigate our body within a space. So, like what the left side is, what the right side is. It also plays a role in sensory information to the cerebellum. It can help with control of the heart rate, vasodilation. If you have like a coughing fit or if you're inducing vomiting, that is all to do with the medulla oblongata that you're looking at. The reticular formation That has to do with alertness and also some skeletal and other muscle types of activity. It will also allow you to kind of filter out repetitive stimuli. So it allows you to kind of focus on key terms. Kind of like when you're in a very crowded room and all of a sudden you hear your name, like someone was talking about you. That, of course, is part of that you're able to focus on an important stimuli. Here is the end part. So you have the cerebrum, which is the large lobe, and then the cerebellum is the one in the back. That one has to do with lots of motor cortexes, visual equilibrium pathways. It helps with balance and posture. So please take your time. I just think it's extremely fascinating to learn about the brain and all its important functions that it contributes. The brain is also protected by meninges, which are layers of protection. For those of you in lab, you've most likely dissected a brain and it had a white layer over it. That is the dura mater, which is the top meninge. And then underneath that, you have the arachnoid matter. And the deepest layer is the pia mater, which tends to be directly embedded within the top portion of the brain. This from a different view, so you can see the dura mater, the thicker one, and these are all meant to protect and help lubricate the brain. The spinal also have these meninges, the same order, dura mater, arachnoid, pia mater. In this illustration, it highlights the fact that the spinal cord is basically an extension of the brain. And as it extends down, it is wrapped around the vertebrate column, protected by the bones within the back. Interesting part about the spinal cord is obviously the nerves will start radiating outwards. So you can see the notation of the cervical spinal nerve, the thoracic spinal nerves, the lumbar spinal, and the sacral. The spinal cord does not run the entire length of your back. It usually stops around L1 to L2. And when it comes to an end, and you can see it right here, when it comes to an end, it will end in what we call the medullary cone or the conus medullaris. And from there, it will radiate out with several nerves, and it kind of looks like a horsetail, so they call it cauda equina. There you go. So the spinal nerve does not run, the spinal cord, my apology, does not run the entire length of the back. Here is a view of the spinal cord not only embedded within the vertebra, but you can also see it with the meninges around it. So they highlight the fact that you have your dura mater, arachnoid mater, and pia mater that come into play. As you get to this particular part of the outline for chapter, it is also going to highlight, and this is very important to realize, that when you look at the spinal cord, there is this distinctive difference between what's happening on the dorsal, the back end, versus the ventral, the front end of the spinal cord. The dorsal part will be involved with responding to the sensory system. So you can see here in the coloration of blue and green, you can see that it talks about the use of receiving inputs from the sensory neurons, whether it's somatic or visceral. And then the ventral section, the front section of the spinal cord, is going to be all about responding. So that's going to be here highlighted in yellow and red. We can see our look at the fact that it is responding to either the autonomic or visceral or the somatic motor neurons. So please keep that in mind, that the dorsal is for sensory and the ventral is going to be for the afferent or the response. You will also read a little bit more about ascending and descending tracts. These are going to take a look at how the message is spread between the dorsal and the ventral, allowing once again for the nervous system to highlight its communication skill by allowing for a stimuli from the sensory system to be integrated by the interneurons, and then the effect to make its way down to the effector, whether it's the somatic or motor neurons that will play a role in the ultimate response that is needed. All right, having said that, I'm going to stop here. Please keep in mind that it was just a really basic overview or summary of chapter 11 and 12. Please take your time and read through the chapter outlines. And if you have any questions, comments, or concerns, please do not hesitate to reach out. Thank you and goodbye.